One of the great things about the CEM lecture series is we get to hear a variety of viewpoints, and in particular, I think the last one was on mosquitoes and resistance, and today we're completely shifting, and that's part of the fun of this lecture series. So today I am pleased to introduce Dr. David Enard. He is an assistant professor at University of Arizona, down the road, and he's been there for a year and a half. He did his PhD at the ENS in Paris, and then a postdoc at Stanford with Dmitry Petrov. So, welcome.
Thank you. Thank you very much for the introduction and for the invitation. I've had a really good time so far talking with the different people I've met. Today, I'm going to talk to you about what you can do if you want to study ancient epidemics and ancient pathogens, but you actually don't have access to molecular remains from these ancient pathogens, whether they are viruses or bacteria, but I will focus more specifically today on viruses. So what you can do is actually use signals that have been left by ancient pathogens in the genomes of their host during arms races, so coevolution of resistance of the host or tolerance of the host to their pathogens. And if you think about it, it's really like if you were studying, like for example, you were a paleontologist who studies dinosaurs, doesn't have access to the bones, fossilized bones themselves, but finds footprints, fossilized footprints in the mud. And you can still say things about those dinosaurs. So that's very similar. It is also important in the case where you have pathogens where you actually cannot get molecular remains. So if you think of like bacteria, like the case of the Great Plague, we have sequenced DNA from Yersinia pestis. It's pretty recent. You can still find these remains in molecular remains in graves, medieval graves. And so in this case, you can have direct evidence. For a lot of pathogens, you're actually never going to be able to recover such remains. For example, there's a whole family of RNA viruses whose genomes are encoded by RNA. RNA is much, much less sturdy than DNA, and so there's no way, for example, or it would take a very, very big technological leap if to be able to find, like say, 57-year-old RNA from 57-year-old pathogens. So what you can do instead is using adaptation in those host genomes. And probably most of you are more familiar with cases like antiviral proteins and adaptation in those antiviral proteins. So it's typically the kind of work that has been done by people like Sarah Sawyer or Amit Malik, where you have a strong prior to expect adaptation in antiviral proteins in response to different viruses. And it's really a case where you expect the antiviral proteins to adapt so that they can attack new pathogens, new viruses in this case, better than they could in the past when epidemics started. If you had only adaptation in like specialized antiviral proteins like this, it's not enough to be able to say anything about which specific viruses, in this case, which specific families of viruses infected hosts in the past. What you have, though, in the case of viruses is that virologists have been working a lot in the past, let's say, 30 years to identify interactions between different viruses and the host proteome. So in the case, the human host. And if you look at the number of interactions that virologists have found so far, it's actually a staggering number of interactions and a very, very significant proportion of the human proteome that is known to date to interact with different viruses. So if you look at low throughput methods to identify interactions between viral proteins and host proteins, It's like communoprecipitation, GST pull down, and so on. You've got to date actually something close like 2,000 human proteins known to interact with viral molecules. If you add to this interaction that has been found by high throughput methods, such as high throughput mass spectrometry, it goes up to like 4,500 or so interactions, and that actually represent 20% of the human proteome believed known to interact with viruses. And if you look at the distribution between different viruses and infect humans, you can have pretty substantial numbers of interactions with each of the different viruses. And usually the differences in the number of interactions for each virus just really represents the effort of research that is dedicated to different viruses. So not very surprisingly, influenza virus and HIV are the two viruses where there is the most research done, and so the highest number of interactions that have been uncovered. So this high number of interactions is going to be very important if you want to detect ancient epidemics. And the reason why it is important is that if you want to detect ancient epidemics, through the point of view of host genomes, you basically need statistical strength in numbers. What I mean by this is that imagine you know only 5 genes in the human genome that interact with a specific virus. Then you might have one adaptive event in those 5 genes, but one adaptive event out of all the adaptation that has happened in the human genome is not going to be enough to be able to assign this signal of adaptation to a specific virus. So here is how it would work. You can imagine that different pathogens, here you have the case of hypothetical virus A, hypothetical bacteria B, and you know where on the host chromosomes the loci of interactions are located. And of course, you're going to have a lot of those interacting loci where you don't see any adaptation. You're also going to have a lot of adaptation in the host genomes that have nothing to do with pathogens and might be related to adaptation to new diets, adaptation to climate, and so on. But for the specific interaction with virus A or the specific interaction with virus B, you're going to be able to count how many of these different loci have signals of adaptation in the genome at different evolutionary times. And so you might be in a case like this one, where you can see for virus A that a lot of the loci known to interact with virus A in the host, across the host chromosomes actually have signs of signals of adaptation. Whereas for, in this case, bacteria B, you don't have that many signals of adaptation. And in the case of virus A, you might be able to statistically see that you actually have a significant enrichment in adaptation at those loci that is compared to the rest of the genome. So everything I'm going to talk about from now on, when I talk about enrichments and of adaptation at genes that interact with specific viruses or with viruses in general, it always means enrichment compared to controls in the rest of the genome. And I'm going to call genes that interact with viruses VIPs for virus interacting proteins. And non-VIPs in this case are all the other proteins that are not known to interact with viruses. And for the rest of my talk, what I'm going to do is that I'm going to show you comparisons where you have estimated amounts of adaptation in VIPs compared to non-VIPs. And when I compare VIPs and non-VIPs, I always do it in a way that controls for all the other things that could impact adaptation. So for example, the virus interacting proteins that I was just talking about, they tend to be much more expressed, much more highly expressed in different human tissues than non-VIPs. And if you want to look at an enrichment and of adaptation in VIPs compared to non-VIPs, then it's a problem, because it could be that in general, higher expression is connected to higher rates of adaptation, and VIPs might have more adaptation just because they are more expressed, rather than because of the fact that they interact with viruses. And then you might confound something that is just related to how adaptation works in the hosts with the impact of interaction with viruses on adaptation. So everything I'm going to show has always been done controlling for a lot of those controlling for a lot of those confounding factors. So if you think about what was a good time in human evolution to look at the impact of the footprint of ancient epidemics in genomes, it's really hard to come with a better time than the time of interbreeding that happened between modern humans and Neanderthals. Because if you think about it, they had evolved for a while isolated from each other, with modern humans in Africa and Neanderthals outside of Africa. And they evolved in isolation for long enough that they probably had their own pathogens that they were infected with. And it's really not a stretch to imagine that when modern humans and Neanderthals came into contact and started interbreeding, it's really not a stretch to imagine that they gave each other whatever respective STIs that they had at the time. So the idea then is that if they infected each other with STIs, and in particular with viruses, you probably should be able to detect this as in the form of adaptive introgressions in their genomes. And in particular, what we have now from having sequenced a bunch of Neanderthal genomes is that we know in the modern human genomes, so imagine here that the pieces of modern human genomes are in gray. We know that Neanderthal DNA introgressed into modern human Eurasian populations. So imagine modern human pieces of genomes are in gray, and Neanderthal DNA is in green here. What you would have at the beginning of the interbreeding and soon after the interbreeding is that if these pieces of Neanderthal DNA were adaptive against the pathogens that the Neanderthals had infected us with, had infected Eurasian individuals with, then you would have an event of what is called adaptive introgression. So what happens with adaptive introgression is that this stretch of Neanderthal DNA that is at a very low frequency at first, because of the selective advantage it conferred against a specific virus, in this case coming from Neanderthals, it is going to increase in frequency pretty fast, faster than recombination, as actually time to break it and recombine it with the gray modern human DNA. So it's really a case of what you might have heard about, like partial sweeps, where you have a new mutation and then the new mutation increases in frequency together with its carrying haplotype. It's very much the same dynamic here. And if you compare it to what you expect if you have a neutral stretch of Neanderthal DNA, this stretch of Neanderthal DNA, because it's neutral, it's just going to hang around in the population, but it's never going to increase so fast in frequency as an adaptively introgressed Neanderthal segment would do. So that's right after the adaptive introgression. But of course, the interbreeding between Neanderthals and modern humans happened quite a while ago. It happened 50,000 years ago. So what happened in these 50,000 years is that this block of like clean and visible Neanderthal DNA, easy to detect Neanderthal DNA if you were to do population genetics analysis right after the interbreeding, becomes more difficult to detect. And it's more difficult to detect because in the past 50,000 years that occurred since interbreeding, you had a lot of recombination happening, and also you had a lot of erosion, either because of drift or because of purifying selection in some of those pieces of Neanderthal DNA were containing deleterious fragments. And so what we found at the time, I'm not going to get into the details of this specific part of my work because you already apparently studied the paper, is that what we found that these cases here of like adaptive introgress segments from Neanderthals to modern humans were much more likely to overlap specifically in European modern human populations. They were much more likely to overlap proteins that interact with RNA viruses as opposed to proteins that interact with DNA viruses. And so a way you can interpret it is that it makes it likely then that the viruses that Neanderthals infected us with were more likely to be RNA viruses rather than DNA viruses, at least for the ancestors of present modern human Europeans. We actually didn't find any evidence of this enrichment in East Asian populations today. So it could have been that it was like there were multiple bursts of like interbreeding between modern humans and Neanderthal. So it could have been an episode of interbreeding that happened specifically with the ancestors of modern human Europeans. Or it could have been that the enrichment would have been visible also in East Asians, but there could have also been further enrichment of adaptive introgression driven by a DNA viruses specifically in East Asians. And that would have like put the balance back in East Asian populations. That's really hard to tell, and that's something I'm working on. So that's something that happened 57 years ago. And like I said, it was just a particularly good time to start studying ancient epidemics because it made so much sense that at this particular time in evolution, you would expect exposure to new pathogens, basically. You can start looking at ancient epidemics then in a like more blind way and just like look at any signal of adaptation at different evolutionary timescales that you can possibly use and that we know how to use in population genetics. So one thing I did after was to focus on more recent human evolution, and I just use human populations in case from the 1,000 Genomes project. So it's 26 populations. It's still very far from representing the whole diversity of the fine scale diversity of human populations, but that's a start. And what I did specifically was to look for signals of recent adaptation called selective sweeps. So for those of you who don't know about selective sweep very well, I'm just going to describe now how it works. So imagine you have an adaptive mutation that happened on a specific chromosome in a population, and that this population has, of course, genetic diversity, here represented as like neutral variants that are around the new adaptive mutation. And these neutral variants are found in different combinations in different chromosomes. And these combinations of different variants each define what is called haplotypes, and like specific haplotypes that are represented here in different colors. So this adaptive mutation is adaptive, which means carriers of the mutation have more progeny. And if it is strongly advantageous enough, what happens is that in a population, the frequency of the adaptive mutation increases fast enough that recombination doesn't have time to recombine this particular gray haplotypes here that carries the adaptive mutation together with the other haplotypes in the population. And what you see happening is that because this haplotype increases in frequency together with adaptive mutation, it leads to loss of diversity around the adaptive mutation of like neutral diversity. So that's for what is called a partial sweep or incomplete sweep, when the adaptive mutation is not at 100% frequency yet. But once the adaptive mutation has reached 100% frequency, you can see that around it you have a pretty severe loss of neutral genetic variants. So literally, neutral genetic diversity has been swept away around the adaptive mutation. And this is why it's called a selective sweep. So what I did more specifically in this case to look for recent adaptation in response to viruses was to focus specifically on incomplete sweeps. So incomplete sweeps represent pretty recent adaptation. And in the case of the statistics I use, we know that the statistics only have power for the past 30 to 40,000 years of modern human evolution. So what I did first was to compare VIPs with control non-VIPs. So if you remember, controlling for a lot of different confounding factors. And I'm just going to detail what this graph means here. So what I did specifically to measure the enrichment is that I measured the enrichment as the in adaptation as the number of sweeps in VIPs compared to the number of sweeps in a lot of control sets of non-VIPs. So what you see here as the y-axis is the ratio of the number of sweeps at VIPs, proteins that interact with viruses, compared to the controls that don't interact on the number of sweeps in the controls that don't interact with viruses. And the x-axis, basically, what it means is that, for example, if you take the top 200 X position, it means that I looked at the number of sweeps in the top 200 sweeps in the whole genomes, given the strength of the statistic I was using, the strength of the evidence for the presence of a sweep at a specific location in the genome. I looked at this number of the top 200 in VIPs, and I looked at the number among the top 200 in non-VIPs. And the specific statistic that I use, I forgot to mention, is called IHS for Integrated Haplotype Score. It's a pretty standard statistic now that specifically detects long and frequent haplotypes. Long and frequent haplotypes as what you expect only if you have a strong selective sweep. So where a haplotype has increased in frequency fast enough, that recombination didn't have time to like chop it down in into smaller pieces. And so what we saw comparing VIPs and non-VIPs in this case was that indeed you have a strong enrichment in recent adaptation at loci that interact with viruses. And this is worldwide. This is combining sweeps from all the 26 populations from the 1,000 Genomes project. And what is interesting here is that, yes, you see a very strong enrichment in the most extreme sweep signals at VIPs in the human genome. But you have this tail here where you have like something like a 30–50% enrichment in much more lukewarm signals of selective sweeps, which would qualify more to be described as polygenic adaptation. So in that case, that would be like more polygenic adaptation that has still left signals of hitchhiking that are just much more moderate than if you look at like what the top 50 sweeps in the human genome are. So now what that means is that we have potentially a lot more sweeps happening in response to viruses that would be expected by chance, that is expected compared to the rest of the genome. And so it means that potentially we have a lot of statistical power to actually go try to say which viruses drove the signal in specific human populations. And so what I did first was to cut my data set of VIPs in half, with half of the VIPs interacting with RNA viruses and half of the VIPs interacting with DNA viruses. So it's just like a fortunate property of the data set that actually we know that half of the VIPs we have interact with RNA viruses, and about half interact with DNA viruses. So which is very nice. The reason why it's really nice is that that means we have the same statistical power to detect a possible enrichment in adaptation in RNA viruses compared to DNA viruses as like two big groups. And so when we looked at it, this is what we found. So basically, what we did in the case of the black curve here was to compare signals of selective sweeps close or at loci that interact with RNA viruses versus far from any interaction with RNA viruses. And what we did for the orange curve was to compare regions of the human genome close to DNA viruses compared to all the other regions far from DNA viruses. And what you can see is that what really matters is not really if you are close or far to DNA viruses. Basically, there's the same amount in the DNA VIPs and their controls. What really matters, apparently, is how far you are from an interaction in the human genome with RNA viruses. So at this point, you really start seeing, I mean, it's starting to be a pattern, right? It's like RNA viruses we saw were apparently more important at the time of interbreeding with Neanderthals. And again, here we see RNA viruses having a much, much stronger impact than DNA viruses during more recent human evolution. And this was again worldwide. What we did next was to try to find more specifically which viruses and where those viruses like left these signals. So I looked at all the different RNA viruses I had in the data set of VIPs. And quite surprisingly, the strongest signal by far is a signal for VIPs that interact with HIV outside of Africa. So what is confusing here is that if an ancient retrovirus that was using the same interactions with human cells as HIV does today, we know these lentiviruses and most of the retroviruses come from Africa. So it is a little bit confusing to find this signal everywhere except where we expect these ancient viruses would have been coming from. But I'm going to come back to this later. Another signal that we found that is this time more in line with where we know they should have happened is a signal that could have been left by some form of filovirus that was using the same interaction as Ebola virus is using today. And we found this signal all over Africa, both in West Africa and East Africa. But we also found a very, very strong enrichment at those filovirus interacting proteins, specifically in Europe, in Italy. If we look at Northern European populations, that's why I'm not going to look at my video because I know my accent is terrible. And, you know, I keep mispronouncing things, which my wife makes fun all the time. But anyways, so we have this signal that is like very specifically in Europe, in Southern European populations, and more specifically Italian populations. So that means we do have, in this case, geographically defined signals of ancient epidemics, which is what you would expect for at least some ancient epidemics. And what I did in this case is that I checked that those signals statistically were solid signals by doing a very thorough, what is called a false discovery rate analysis, where basically what you do is that you randomize human genomes in a way that makes them look like the real genomes in terms of like clustering of sweep signals with neighboring genes and so on. But otherwise have the locations of where the interaction with viruses happen all scrambled. And we did that. What we found was that there's just no way that these enrichments would have happened by chance. So this is just to show you what the signal with HIV VIPs look like. So this is what the enrichment looks like in Peruvian populations that have been sampled for the 1 Genomes project. What is useful here is that we detect this signal both in Native American genomes and European genomes, and to a lesser extent in Asian genomes. But basically, it tells us the sharing of this signal tells us that probably those signals were generated before the split of Native American populations from their European and Asian ancestors. So that's probably something that happened further than 50,000 years ago. Now that's the signal we have with the Ebola, like filovirus VIPs in Italian genomes. And in that case, this strong enrichment, we don't see it at all in Native American populations. So that's more a case that basically would be more recent than 15,000 years ago. And it's kind of consistent with the fact that we also detect this signal only in very specific European populations that have also diverged from each other not so long ago. The other nice thing about seeing this signal specifically in populations but not in others is that confounding factors that would be shared between those populations cannot possibly explain these signals, because otherwise we would see the signals everywhere. So that's for recent adaptation. Now, what I talked about was just using the IHS statistic. What I'm doing now is that I'm working on creating more powerful statistics to detect incomplete selective sweeps. So what we're going to do in particular in this case, what it shows here is the statistical power of 9 different statistics. The blue ones are existing statistics. The more red ones are statistics I have created recently. And what we're going to do with it is put all these statistics into a machine learning framework to really detect those selective events with a lot of power, to have more power to detect ancient epidemics. And this is work that is being done now by a new postdoc in the lab, Elise Lauterbur, who did her PhD before at Stony Brook University. And what Elise is doing right now is she's training convolutional networks with all these statistics to detect all kinds of selective sweeps with more power than we were able to. And for the story, we've just started using some of those statistics that are able to go fish for positive selection at all the times compared to IHS. And the idea in this case is, I can skip this. If you remember, I was just saying that we have this signal at HIV VIPs pretty much everywhere in the world except where we expected the most in Africa. But if you think about it, it could just be a matter of like the timing of when the adaptation happened and the sensitivity of your statistics to detect adaptation at different evolutionary times. And what we know about IHS is that it's sensitive to adaptation only for the past 30,000 years. If you had something that was older, you're not going to be able to detect it. And if you think about it, given what we know where lentiviruses are today, this is a case where possibly an epidemic could have started in Africa, spread later outside of Africa, and whatever signals that exist in Africa are too old to have been detected by IHS. So what we did is that we scanned the 1,000 genomes again with statistics that detect older adaptive events, and sure enough, we actually find a very, very strong signal, older signal in Africa. And so it really suggests that you had an epidemic starting in Africa, but spreading later in the rest of the world. So it's also consistent with some of the patterns that we see in Europe, where we see the most recent signals for this particular potential epidemic in Northern Europe compared to Southern Europe. So if you imagine a gradient that depends on the proximity to Africa, this is pretty much what you expect. So that's for recent adaptation. So now we have two different times in like fairly recent human evolution where RNA viruses seemed to really dominate the picture when it comes to ancient epidemics. How much time did I talk already? 36, okay. Sorry, I don't have it on the... All right, so that was for recent evolution. Now, if you look at more long-term evolution, more long-term human evolution, you can use what is called the McDonald-Kreitman test to this time look at adaptation since divergence with the common ancestor with chimpanzees. And what I'm going to do now is that for those of you who don't know about the McDonald-Kreitman test, I'm going to describe how it works. So what it does is that instead of just looking at recent adaptation, it really gives you an idea of the amount of adaptation that happened across millions of years. The only problem is that compared to selective sweeps are kind of blind to where or the nature of the mutations that happen. It could have been coding mutations, non-coding mutations. It doesn't really matter. For the McDonald-Kreitman tests, you need to focus on coding sequences of proteins. And what you need is genetic diversity within a specific species, and you need to know what are the differences in the genomes of the species where you are interested in looking at adaptation compared to a closely related relative. So in this case, chimpanzees. So what you need to run the McDonald-Kreitman tests, first you need the polymorphism in the coding sequences, both at synonymous positions and non-synonymous positions. Synonymous positions, they are just polymorphisms, variants, that change the codon, but don't change the codon in a way that it changes the amino acid. So like every amino acid can have different codons that they are coded with in like coding sequences. So sorry, I was pointing to the wrong column. That's the synonymous codons. You also have non-synonymous variants. So those non-synonymous variants do change the amino acid, and because they change amino acids, in a lot of cases, it is actually going to be a problem for the function of the protein. And so those new non-synonymous mutations, a lot of them are going to be removed by purifying selection. And so typically, you see way less non-synonymous polymorphism variants, compared to synonymous polymorphisms. So that's the variance you have in a population. If you look after a long evolutionary time, what you can say is that imagine all those variants a long time ago were neutral. If all the variants were neutral, it would mean both the non-synonymous variants and the synonymous variants have the same probability of becoming fixed, so of reaching 100% frequency in a population, and at this point become a difference between species, right? So if you were comparing humans and chimpanzees, you would have the same probability after a while to see these variants becoming divergent sites between human and chimpanzee. So in this case, what you expect is that if you measure the ratio of the number of non-synonymous sites over synonymous sites under a strictly neutral scenario, you expect this ratio to be equal to the ratio of non-synonymous differences between species compared to synonymous differences. Now, what happens is if you have adaptation, those non-synonymous variants, which are the ones that adapt, you just make the assumption that synonymous variants are never adaptive. Those non-synonymous variants are more likely to become fixed than the synonymous ones. And so in that case, this higher probability of fixations means that you're going to end up with more divergent sites than expected under neutrality. And this is the excess here that is noted A. So what that means in that case is that you're going to see that the ratio of fixed differences between species is higher than the ratio of variance. So you can quantify A. The difference between the two ratios makes it possible to quantify how many adaptations you had. And you can just make it a rate of adaptation just by dividing with the total number of non-synonymous differences that you see. So alpha is like a classic adaptive quantity that you see a lot when it comes to quantifying adaptation that happened in coding sequences. So if it was that simple, I would like not even talk about it, and it would be like pretty straightforward. Unfortunately, it's really more complicated than this. I talked so far about neutral variants and adaptation. You also have a lot of deleterious variants in coding sequences. And of course, some of those variants are so deleterious that they never make it to frequencies high enough that you can really see them in a population. But a lot of those variants, they are more weakly deleterious. And so before they eventually become removed from populations, they can still stay for a while at low frequencies in populations. But what that means is that actually you see them as non-synonymous polymorphisms. And so what it tends to do is that it tends to inflate the number of synonymous variants compared to a case where there would be no deleterious variation. And so this is a problem because it can completely mask the amount of adaptation and actually lead to negative estimates of alpha, which doesn't make sense. You can't have a negative rate of adaptation. The other problem with the magnetic regression test is that not only deleterious variants can inflate PN, you also have a strong assumption about how fast adaptive variants become fixed. There is an assumption that is often made in the magnetic regression test that all adaptation is strong enough, that it's fast enough, that those adaptive variants don't participate to polymorphism and just participate to divergence. But in the human genome, especially where you have pretty small effective population sizes, it's not true. You have adaptive variants that participate to genetic variation. And this is what is shown here. So this is work that I did in collaboration with Lorenzo Riccio when I was a postdoc at Stanford. And Lawrence really did all the analytic part of this work. He's on the job market right now. Just to let you know, he has a lot of, he already got a lot of interviews. He published 6 papers just in the past year. And this is all, pretty much all the analytic part for this project was done by him. So I'm just saying that, but I think he's a pretty good person to consider for, as a young faculty. So what it shows here is that if you look at a curve of alpha as a function of the frequency of variance, you have this big drop on this side, which corresponds to the deleterious variance. Because you have a lot of the deleterious variants that are at low frequencies, their effect of underestimation on alpha is stronger at low frequencies. And what happens here at high frequencies is that when you have weak adaptation, it's slow enough that you have a lot of the adaptive variants that are still present in the polymorphism, as opposed to the case where you only have strong adaptation. And this drop here of alpha at high frequencies corresponds to this weakly advantageous variance. The nice thing is that when you have this shape of this alpha curve, you can actually exploit it to not only estimate alpha, but actually also estimate the strength of adaptation. So that's what we did in the case of VIPs compared to non-VIPs. And to speed things up a little, we can focus here on the difference between VIPs and non-VIPs for all advantageous mutations. So that's the yellow here. And you see the difference is very clearly above 0. So a lot more adaptation happening in proteins that interact with viruses compared to those that don't. And if you look at strong adaptation as opposed to weak adaptation, what is interesting here is that you see that weak adaptation is not so different between, in blue, VIPs and non-VIPs, but strong adaptation is really what drives the excess of adaptation at VIPs. And if you think about like viral epidemics, indeed, you can imagine that it's going to be like very strong selective pressures as compared to like other selective pressures. So now, coming back to like which viruses could have driven this adaptation, this is work that that was just finished by an undergrad in the lab, Carl Jenkins, who just started as a senior at U of A. What we did was to look at adaptation with the tool developed by Lawrence. Again, comparing RNA VIPs and DNA VIPs. And what we saw was DNA VIPs, the difference with the controls is really not there. There's no difference in adaptation between DNA VIPs and non-VIPs that otherwise have the same level of like different confounding factors. For RNA VIPs, instead, there is a very, very large difference. And again, if you look at the difference, it is mostly due to strong adaptation as opposed to weak adaptation. So now it's more than coincidence. There is really, really strong evidence at different evolutionary timescales in human evolution that RNA viruses have driven more adaptation than DNA viruses. And the nice thing about this result is that if you look at the epidemiology of viruses today, pretty much 95% of zoonoses that have been recorded since they are starting to be recorded is actually due to RNA viruses, whether it is HIV or Ebola or the flu or other viruses. Maybe I'm like going over, I don't know. Okay, thanks. So I'm just going to say a few last words about what this adaptation does in these proteins, because it's nice to see a strong enrichment in adaptation in those proteins, but then the question is, what happened to these proteins? And if you think about the classic interpretation that is made, especially with regard to viral receptors, most of the mechanisms of adaptation that are known involve the interface between the viral protein and the host receptor. And the adaptation is often described at the level of this contact interface with the receptor. But there's actually other ways that you can think of that could lead to adaptation against a virus. So yes, it could be a change in the contact interface. So the contact interface, because of adaptation, might be lost for the virus. It's like strategic evasion for the host that has been described in the past. But you can imagine that the host protein activity is modified. Or you can imagine that the stability of the protein is modified. And for example, for a lot of proviral proteins, so that proteins in the host that the virus needs to complete their replication cycle, you could imagine that having less of a specific host protein would then be detrimental to the virus. So what I started doing was to look at where adaptation occurred using 3D structures of proteins, human proteins. And if you look at where, basically, if you ask where with proteins, it is really a matter about whether a specific residue is buried in the structure or if it is exposed at the surface of proteins. And in blue, we have the rate of adaptation in different parts of the proteins. And orange, we have what happens at control non-VIPs. And you can see that if you compare to the classic expectation of like surface adaptation at the contact with the viruses, this is not quite what seems to be happening here. There is actually not more adaptation at the surface at VIPs compared to non-VIPs, at the surface of VIPs. The excess of adaptation is like halfway between the most buried residues and the most exposed residues. And those like parts of the protein that are halfway are pretty much expected for different reasons to be changes that are going to impact the stability of the proteins. So what I'm working on now is to try to show that mostly when you have adaptation in VIPs in response to viruses, it probably involved a change of stability in the proteins. And what we know actually is that most of the VIPs are proviral as opposed to interviral. So we have a strong reason to also test that potentially more destabilizing changes happen compared to stabilizing ones. So that's pretty much what I wanted to say. So this is the kind of work that is done right now in the lab. And if you're interested, please contact me. And thank you for your attention. And sorry for taking a bit more time than I expected. Sorry. Yeah.
Regarding the importance of RNA viruses versus DNA viruses, is that because RNA viruses have an easier time infecting hosts, or they're more prolific, or what are the factors?
So if we look at like the past 100 years, definitely it looks like they jump host species more often than DNA viruses. And DNA viruses tend to be more vertically transmitted, like in evolution, with like cases like HPV, for example, where the evolution of the virus has followed human evolution for a longer time. So there's definitely a difference in the rate of transmission from a species to another. What explains it then at the level of the viruses and how they interact with the host? I would say I haven't done enough homework on that to respond very precisely.
I don't know how much of overlap you have in your RNA virus interacting proteins, but if they're like a small subset, then when you have sort of constant viral interactions, do you think you would end up having balancing selection on those?
That's something I have not tested. Mm-hmm. But you do have VIPs that interact with both RNA viruses and DNA viruses. So those, of course, I have to exclude when I try to differentiate RNA viruses and DNA viruses. Like P53, for example, that is much better known for being a tumor suppressor, is already known to interact with like 20 different viruses or more. And yeah, that's definitely something I have to take into account. There's probably balancing selection going on, especially if you look at VIPs that are immune VIPs. You see that when you run the Maynard-Quetman test, it gives you estimates of alpha that are very low, as if there was balancing selection, increasing PN, especially at like intermediate frequencies.
Yeah.
So following up on that same question, how do you untangle the concept where you'll have dual infections constantly happening, where you'll get adaptations from RNA and DNA viruses at the same time? You do not have one or the other. You'll have both of them at the same time.
Yeah. So if you have this overlap during evolution, it is basically, in our case, it's going to limit the power we have to discriminate the signals. if it was the case during all the time during evolution, there would be the same amount of adaptation at DNA VIPs compared to RNA VIPs. The fact that we see a difference probably means that at least for a significant amount of evolutionary time, those things happen separately enough.
All right, can we thank the speaker?
Thanks.